Welcome to this video on stem cells, created for your GCSE Biology revision. We will cover the definition of stem cells, how they differentiate, and their importance in medical science. In this video, we have several learning objectives. We will define stem cells, explain their role in differentiation, compare different types, and evaluate their potential use in medicine, along with the ethical considerations. Before we dive in, consider these questions. What makes a cell specialized? And can a single cell produce different cell types with vastly different structures and functions? What is a stem cell? Simply put, they are unspecialized cells that have not yet developed a specific structure or function. Unlike other cells, they can divide and differentiate into many types. Unspecialized cells lack the specific structures of mature cells. Their future role is determined by a process called differentiation, which transforms them into specialized cells as needed. Stem cells are defined by their ability to divide. This division increases the number of available cells. Some remain stem cells to continue dividing, while others differentiate. This division happens through mitosis, which produces genetically identical daughter cells. It is this process that allows the body to generate new cells for later differentiation. Differentiation is the process by which an unspecialized cell becomes a specialized one. This is determined by which genes are switched on, allowing the cell to develop specific structures for its role. The process happens in two steps. First, division through mitosis, and second, differentiation, where cells receive instructions to change into specific functional types like nerve or muscle cells. Examples of specialized cells include red blood cells, which transport oxygen, nerve cells, which carry impulses, and muscle cells designed for movement. All originate from stem cells. When comparing potential, embryonic stem cells are highly versatile and can form almost any cell type. Adult stem cells are more limited, typically producing cells related only to the tissue they are in. Embryonic stem cells are sourced from early embryos. Because they have not yet adapted for a specific function, their broad potential makes them invaluable for scientific research. Their ability to form a wide range of specialized cells, such as muscle or nerve tissue, means that research into human development often relies on the versatility of embryonic stem cells. By studying how these cells divide and differentiate, scientists hope to gain a deeper understanding of development, potentially leading to breakthroughs in medical treatments. Finally, adult stem cells are found in specific body tissues. Their role is to replace or repair damaged cells within that specific tissue, which is why their potential is more restricted. Moving on to adult stem cell limitations, adult stem cells are restricted compared to embryonic ones. They generally form a narrower range of cells, tied to the specific tissue where they reside, whereas embryonic stem cells have much broader potential. Bone marrow is a key location for adult stem cells. These cells continuously divide to produce new blood cells, such as red and white blood cells and platelets, replenishing them throughout a person's life. The process of producing new blood cells involves bone marrow stem cells dividing to increase their numbers, followed by differentiation into specialized types like oxygen-carrying red blood cells or immune-supporting white blood cells. Because they can replenish blood cells, bone marrow stem cells are used in medical treatments to help replace damaged cells, potentially assisting in cases like certain types of leukemia. Plants also have stem cells, found in regions called meristems. These are located in areas of active growth, such as root tips and shoot tips, and contain unspecialized cells capable of differentiating into specialized plant cells. Root meristems enable downward growth into the soil, while shoot meristems allow for upward growth toward light. In both locations, actively dividing cells continuously generate the new tissue a plant needs. A unique feature of plants is that their stem cells can differentiate throughout the entire life of the plant. Unlike most animal stem cells, which lose this ability early on, these meristem cells act as a source for all specialized plant structures. They produce cells for roots, stems, and leaves, ensuring the plant can develop and function effectively as it grows. To summarize, embryonic stem cells provide broad potential for many cell types, adult stem cells are tissue-specific, and plant stem cells allow for lifelong growth and differentiation in meristem regions. Regarding medical applications, stem cells hold potential for replacing damaged or diseased cells. Research is ongoing into how we might use them to repair tissues and treat various conditions. Replacement works by using stem cells to replenish damaged tissues. Once they are guided to differentiate into the specific type of cell required, they can help restore essential biological functions. Beyond just single cells, scientists are investigating how to produce organized replacement tissues. The challenge lies in controlling the differentiation process to ensure the new tissue functions correctly. Current successful treatments include bone marrow transplants for blood conditions. Future research aims to extend these successes to other areas, such as repairing damaged nerves or muscles. Overall, the study of stem cell development is vital for modern medicine. By understanding how these cells function, scientists move closer to developing innovative ways to treat complex diseases. Finally, we must consider risks. Stem cell therapies can lead to uncontrollable cell division, potentially forming tumors. Therefore, the medical benefits must be carefully weighed against these significant health risks. A significant challenge in stem cell therapy is immune rejection. The immune system may recognize transplanted cells as foreign, triggering an immune response that can damage or destroy them. Careful patient treatment planning and matching donor cells are essential strategies to reduce this risk. Beyond clinical risks, embryonic stem cell research involves important ethical concerns. Perspectives vary on the moral status of the embryo, and these must be carefully balanced against the potential medical benefits in any research or treatment decision. A primary ethical concern is that obtaining embryonic stem cells often results in the destruction of an embryo. While some consider this unacceptable, others argue that the potential for life-saving medical treatments outweighs these concerns. Fortunately, stem cells can be obtained from other sources. Adult stem cells are found in various body tissues, helping with repair and maintenance. Additionally, plant stem cells are found in meristems, where they drive growth in root and shoot tips. When evaluating stem cell applications, we must weigh the benefits, such as replacing damaged tissues and treating serious disorders, against the risks, including tumor formation, immune rejection, and various ethical dilemmas. For your GCSE exams, you may be asked to explain how stem cells are used in medicine. Remember to mention cell division, differentiation into specialized cells, and their role in replacing damaged tissues or producing replacement tissues. When structuring your answers, start by defining stem cells as unspecialized, then explain the process. They divide through mitosis and differentiate into specialized cells to repair damaged tissues and restore function. Let's review. Stem cells are unspecialized, found in tissues like bone marrow or plant meristems. Embryonic research is controversial because it often involves the destruction of embryos, which some view as morally problematic. A key exam tip: always link stem cell division with differentiation in your answers. Mentioning both processes ensures you cover the full scope of how stem cells function, which is critical for gaining full marks. To wrap up, consider these final questions. Which stem cells have the widest ability to differentiate? How does bone marrow produce new blood cells? What are the main ethical concerns we discussed?